Next, we'll be using the story of the development and regulatory history of a single drug called Bevacizumab, whose brand name is Avastin, to show the process of drug development and FDA approval in context. It was Judah Folkman, a young cancer surgeon in Boston, who observed in the 1970s that the malignant tumors he removed were hot, red, and bloody. Folkman reasoned that there must be a substance in these cancers that somehow generated blood vessels to nourish the growth of a tumor. To demonstrate that this observation was real, he injected a tiny piece of tumor tissue into a rabbit's cornea, which normally has no vasculature. Within a day, blood vessels grew into the cornea to nourish the tumor tissue. Folkman called this process angiogenesis. There were many years of resistance in the scientific community to the theory that tumors could be blocked by limiting their blood supply. After frustrating early efforts to isolate the substance, one of the principal genes responsible for angiogenesis, VEGF-A, or vascular endothelial growth factor A, was identified and cloned by Genentech scientists. In 2004, 33 years after Judah Folkman first published his theory in the New England Journal of Medicine, the first angiogenesis inhibitor on the market, Bevacizumab, brand name Avastin, was approved by the FDA for the treatment of metastatic colorectal cancer in combination with chemotherapy. There were safety concerns from the start. Because of its mechanism, Avastin could cause serious bleeding, intestinal perforations, and interfere with wound healing. But these serious side effects seemed to affect only small numbers of patients. The company thought that Avastin would work for all solid tumors that depended on angiogenesis, so they initiated many trials. But the randomized trial aiming for approval of Avastin in metastatic breast cancer failed. Not only did patients not live any longer with the drug added to chemotherapy, but adding Avastin did not even prolong the time before the tumor grew again, known as progression-free survival. In 2006, Avastin was approved for metastatic non-small cell lung cancer, and in 2009, it was approved for kidney cancer and glioblastoma, a primary brain cancer. None of these results were as dramatic as researchers had hoped, but for patients with deadly cancers for which few treatments existed, like colorectal, lung, and the other cancers, these small improvements were enough to secure FDA approval. But this was not the end of the story of Avastin in breast cancer. Some influential researchers refused to give up on Avastin for metastatic breast cancer. Maybe the trial had failed because Avastin had been paired with the wrong chemotherapy drug, or maybe it had been given too late in the course of the disease. ECOG, the Eastern Cooperative Oncology Group, one of the large academic consortia organized to rapidly enroll important clinical trials, was persuaded to sponsor a trial they called E2100. In this randomized controlled trial, Taxol, a standard first-line chemotherapy treatment for metastatic breast cancer, would be compared with Taxol plus Avastin. Ordinarily, a Phase 3 trial would mask or blind the investigators and patients to which drug the patient was receiving through the use of a placebo as a comparison to Avastin. But this trial was open label, meaning everyone knew who was getting the Avastin and who was not. The side effects of Avastin were unique enough, the investigators believed, that doctors and patients would have guessed anyway. But lack of masking can introduce bias, because people may experience what they expect to experience, and doctors may evaluate patients taking Avastin differently than they would if they were not taking Avastin. The early results of this trial were presented to an enthusiastic crowd at the annual meeting of the American Society of Clinical Oncology in June 2005. As in the prior trial in breast cancer, there was no improvement in overall survival when Avastin was added, meaning that patients didn't live any longer, but there was a 5.5-month improvement in progression-free survival, the time it took their cancer to spread. These findings led to many questions. Was this extra time to progression real? And if so, how meaningful was it to patients? Did the serious side effects associated with Avastin detract from the value of the extra time before the cancer progressed? How could there be an improvement in progression-free survival but not in overall survival? Reasoning that other breast cancer drugs had been approved by the FDA before without demonstrating an overall survival benefit, Genentech, the manufacturer, submitted an application to the FDA for approval of Avastin in metastatic breast cancer based on the ECOG E2100 trial. After requiring an independent review of the trial data, the FDA looked to one of their advisory committees, the Oncologic Drugs Advisory Committee, or ODAC, to advise them whether or not the drug should be approved. ODAC's members are practicing physicians, researchers, statisticians, and other research experts as required, as well as consumer and patient representatives and a non-voting industry representative. At the ODAC meeting, the company made a presentation supporting the approval of Avastin. Then the FDA made a presentation focused on their concerns about the conduct of the trial, the acceptability of the progression-free survival endpoint for approval, and the toxicity of the drug. They were also concerned about missing scans and patient follow-up data, and bias from an open-label design. While delaying the progression of advanced cancer sounded as if it would offer a clinical benefit to patients, that might depend on whether that time was spent dealing with difficult side effects. The data on quality of life were incomplete, so reported toxicities were all that could be used to judge how patients were feeling. The FDA found that the addition of Avastin led to a 20% increase in severe to life-threatening grade 3 to 5 side effects. Grade 3 side effects are described as severe, grade 4 as life-threatening or disabling, and grade 5 side effects are fatal. The FDA attributed 6 deaths to Avastin, although this was disputed by the company. The members of the committee were reminded of the lack of progression-free or overall survival for Avastin in the earlier Phase 3 trial in patients who'd already had 1 or 2 lines of chemotherapy. After hearing and deliberating on the testimony from both sides, the committee members split their vote, recommending 5-4 against the approval of Avastin. The FDA is not bound by its advisory committee recommendations, although in practice it usually does follow them. But this was an exception, with the FDA deciding in February 2008 to grant accelerated approval to Avastin. By the time Avastin received accelerated approval, the required confirmatory trials were well underway. The first was called Avado, a blinded randomized trial that enrolled 736 women who had not yet received chemotherapy for their metastatic breast cancer in one of 3 treatment arms, a chemotherapy agent called Taxotere plus a placebo, or Taxotere with 2 different doses of Avastin. The results were very disappointing. On average, the women getting Avastin at either dose had only about 1 month or even less extra time until their disease progressed, a finding that turned out to be statistically significant because of the trial's large size, but which fell far short of the extra 5.5 months progression-free survival in the E2100 trial that had led to Avastin's accelerated approval. The clinically significant side effects were much higher in the Avastin arms of the trial. The question had to be asked, was a few weeks longer before the cancer progressed, really worth the added toxicity of the Avastin. In the other confirmatory trial, known as Ribbon 1, patients could receive one of 3 different chemotherapies depending upon prior treatment. That resulted in a 4-arm trial of 1,237 patients. Here again, there was a small, statistically significant improvement in the primary endpoint of progression-free survival, ranging from 1.2 to 2.9 months with the addition of Avastin to chemotherapy. And again, the grade 3 to 5 side effects were increased in the treatment arms that were randomized to Avastin. In July 2010, another meeting of the Oncologic Drugs Advisory Committee was called to discuss whether the magnitude of clinical benefit outweighed the risks in these 2 confirmatory trials. Should Avastin be offered a full approval based on these data, or should the indication in metastatic breast cancer be withdrawn? Once again, both sides presented their arguments, and the committee asked questions. When the vote was taken at the end of the day, the committee voted unanimously against conversion of accelerated approval to full approval for Avastin. A second vote of 12 to 1 supported the withdrawal of accelerated approval for Avastin in breast cancer. If denied, Avastin would remain on the market for lung, colorectal, and other cancers for which it had received approval, and could be prescribed off-label for breast cancer patients. However, many believed that it was unlikely that Medicare and other insurers would reimburse for the drug in that circumstance. The response was immediate. Patients who believed Avastin was helping them feared losing access. "This is horrific," said Pat Howard, who credited the drug with putting her cancer in remission. "This is like saying, we don't value your life. I'm shocked. I'm totally shocked. Without Avastin, I'm going to die." Some oncologists said FDA had exaggerated what were manageable toxicities and expressed alarm that a treatment option would be removed from their armamentarium. "The concern is that FDA removes the current label. We may be throwing out the baby with the bathwater," said oncologist Kathy Albane. Other doctors faulted FDA for inconsistency, pointing out that other breast cancer drugs had been approved on the basis of minimal but statistically significant progression-free survival and not overall survival. "There has not been a single metric by which the FDA has reviewed drugs for advanced breast cancer," said Harold Bernstein of the Dana-Farber Cancer Institute. Some media saw this as a regulatory blow to industry that would discourage drug development. A Wall Street Journal editorial referred to the ODAC members as government-anointed medical patriarchs, substituting their own subjective view of Avastin's risks and costs for the value that doctors and patients recognize, and went on to claim that what they termed the Avastin mugging was really an attempt to undermine regulatory modernization, like accelerated approval, that offends the FDA's institutional culture of control and delay. But others supported ODAC's vote. "It was a reasonable decision based on scientific evidence," said the New York Times editorial page. "The drug has failed to extend lives in clinical trials, and it carries a small risk of devastating, sometimes fatal side effects. I am as disappointed as anyone else to not see a survival benefit with bevacizumab, but I am willing to be honest about the fundamental implications of the data," said oncologist and researcher Matthew Ellis. "I believe we have to set the bar high for cancer drugs, even in the metastatic setting." Others cited the high cost of Avastin, which could range close to $100,000 annually, and stressed the importance of knowing which patients responded to Avastin, pointing out that the trial results would be more impressive if these patients could be identified and studied. Although I believe there are subpopulations that get substantial benefit and do particularly well with bevacizumab, I don't know how to select those patients," said Eric Weiner. In this era of increasingly targeted therapies, it is our responsibility to identify populations that will benefit the most from new agents. When FDA announced their decision to withdraw the Avastin indication 6 months later, Genentech and its parent company Roche refused to voluntarily withdraw Avastin for the first time since the law that put accelerated approvals into place nearly 20 years earlier. In a tightly reasoned legal brief, the company disputed FDA's decision and requested the further hearing they were entitled to by law. In the interim, Avastin continued to be used in the treatment of metastatic breast cancer, although not as widely as before the FDA meetings. Taking all of its cancer indications together, that is for lung, colorectal, brain, renal, and breast cancer, Avastin remained the largest selling drug in all of cancer treatment, netting a projected $7.5 billion in 2011, down from a high of nearly $10 billion. The company experienced a victory in March 2011 when the EMA, or European Medicines Agency, the FDA's counterpart for the European Union, ruled in favor of Avastin for the treatment of metastatic breast cancer. By the time you listen to this module, a decision will have been made about a continued approval of Avastin in first-line metastatic breast cancer. The Avastin hearing of 2011 will have become just another footnote in FDA history. Patients joined by some advocacy organizations will have pleaded to retain Avastin's breast cancer indication for themselves and for future patients who might benefit. Other advocates and some consumer advocacy organizations will have agreed with FDA that Avastin's harms outweigh the benefit, and that settling for minimal increases in progression-free survival in unselected populations will not advance the science or ultimately make meaningful changes for patients. In the near future, at least, it is likely that the FDA's decision in this case will have an impact on accelerated approval and on drug development. Now that you know a little of the history of FDA, you will be able to see this decision in context. When you read about FDA approval and withdrawal of approval, advisory committee votes, and other regulatory actions, think about all the dynamics of drug regulation that are in play and what they mean. An interesting parallel to the Avastin story in breast cancer has to do with its use off-label to treat neovascular age-related macular degeneration, a common eye disorder in the elderly that results in blindness caused by the overgrowth of blood vessels in the center of the retina. Years before Avastin was FDA approved for colorectal cancer, ophthalmology researchers found that angiogenesis was implicated in this form of macular degeneration. So Genentech developed a treatment, ranibizumab, brand named Lucentis, that was approved in 2006 by the FDA for this indication. Before the approval of Lucentis, eye doctors started offering their patients very small amounts of Avastin off-label, since it is a monoclonal antibody that is very similar to Lucentis. Both drugs are injected directly into the eye. In small studies, Avastin seemed to work extremely well. For this use, in these amounts, Avastin is far cheaper than Lucentis. After Lucentis' approval, Genentech attempted to stop eye doctors from using their own competing drug, Avastin. They tried to prevent compounding pharmacists from making the tiny doses used for injection. Eye doctors protested, stating that many patients could not afford Lucentis. Genentech also refused to conduct a head-to-head trial comparing the effectiveness of the two drugs. So the much-needed trial was funded by the National Eye Institute and the American taxpayer. The results of the CAT trial, Comparison of Age-Related Macular Degeneration Treatments trial, were published in the New England Journal of Medicine in April 2011. The authors of the trial concluded that the two drugs were equivalent in their effect on visual acuity, how well we see, for the first year of follow-up. The authors went on to discuss the issue on everyone's mind. One of the many factors that contribute to selection of a drug for a patient is cost. A single dose of ranibizumab costs 40 times as much as a single dose of bevacizumab. This cost differential has important economic implications when extrapolated to the more than 250,000 patients who are treated for neovascular age-related macular degeneration annually in the United States. The example of Avastin highlights the complex dynamics of cancer drug development and delves into many aspects of the FDA's role and responsibility as a regulatory agency. It also raises questions about the misaligned incentives that persist in the pharmaceutical industry in the context of targeted therapies and personalized medicine. The drug industry worldwide is an unqualified financial success, second only to the energy industry in profitability. But there is a dilemma. While company profits are maximized by marketing drugs to the largest number of patients, clearly benefits to patients are maximized by treating only the right patients at the right time with the right drugs. Overtreatment and overuse of drugs has become a major concern in the United States, and since drug companies strive to maximize sales, financial incentives for research do not favor development of biomarkers. Yet there are some favorable tools that spare those unlikely to respond to a given treatment the toxicity and cost. By keeping standards high for drug approval and requiring meaningful clinical benefits, such as survival improvement measured in months and years in cancer, the FDA can encourage early efforts to co-develop these tests along with drugs. By selecting a more responsive population to test, clinical trials could be smaller, and minor improvements that are diluted in a general patient population could be transformed into true clinical benefit of real magnitude in those who do respond, thus sparing others. Some fear, however, that raising the bar will simply diminish market size and discourage companies from investing in drug development. Others fear that the inherent conflict between market share and targeted therapy will move us towards ever more expensive designer drugs. Whatever happens, it's clear that the FDA will play a key role in the process.